And I will start the live stream. And I believe we are live. Welcome everybody to another session of the Sussex Vision seminar series. And it's a part of the worldwide Neuro initiative, which was established by Tim Vogels and Panos Bourselos. I'm Marvin Seifert, PhD student in the lab of Tom Baden at Sussex University. And I'm delighted to host today's guest, Professor Dr. Herwig Baier, who is a director at the Max Planck Institute of Neurobiology in Munich. Herwig studied biology at the University of Constance in Germany and did his PhD with Friedrich Bonhoeffer at the Max Planck Institute for Developmental Biology in Tübingen. He then did a postdoc in the lab of William Harris at the University of California. And he stayed in California and became professor at the University of California in San Francisco from 1998 to 2012. And currently he's a director at the Max Planck Institute of Neurobiology in Munich. This position he has since 2011. And Herwig and his team use quite a diverse area of different methods to study zebrafish vision, or vision in zebrafish mainly. Methods like molecular, genetical, optical, connectomic, behavioral, and computational approaches. And I think Herwig's studies have given quite a sophisticated insight into different aspects of zebrafish vision. And that's why I'm really excited to see what work and what data he has prepared to show us today. And I think with that, I would say the stage is all yours, Herwig, and I think I want to thank you once more that you share your work with us today.
Well, thank you very much, Marvin, for the kind introduction. I'm also looking forward to sharing my data with you. I'm going to share the screen first. Okay, so it's a pleasure to be speaking in this series, which I've been following for a few months now. And I'll gladly talk about our data. I hope there's going to be some audience there, or everyone is tired of all the Zoom talks and Zoom meetings. But it's going to be, if I understand correctly, on YouTube, so you can also check it out later and tell your friends. So I will talk about our work on the visual system of zebrafish, and I'm going to keep it, or try to do it in an interesting fashion by being somewhat provocative in my statements, in my conclusions from this work. Because I think that our work on this vertebrate visual system has really changed, at least my view of how vision works. And I'm going to kind of guide you through the fish visual brain, and I'm going to go from molecules to behavior. So the first video that I'm going to show you was not taken by us. I lifted it from Wired magazine. Someone took the pains of filming fish from below. So here's a minnow in a lake, and which is frolicking happily in the water. Unbeknownst to this little fish, there is a looming predator that is going to snatch the little fish out of the water. So here's a kingfisher that has successfully caught this prey item and is going to eat it or feed it to his young. So what exactly is happening in this situation? So let's break down the situation for the two visual systems involved here. So that of the kingfisher bird and that of the fish. The bird detects a moving object of the right size from above. The bird categorizes the object as prey. The bird locates the prey. The bird selects one prey against a background of distractors. So maybe some of you saw another fish crossing the field of view. So the bird really has to pay attention to one target and select this and not let himself be distracted. The bird aims his movements to catch the fish. The fish recognizes a looming shadow, so try to escape at the very last minute, in this case unsuccessfully. The fish escapes or tries to, but is not fast enough. But clearly this fish, since it's been around, has had a line of ancestors that were quite successful in avoiding being caught. The bird catches the fish. So if we summarize and try to derive some basic principles from these observations, we can conclude that successful behavior, be it hunt or escape, requires object detection, classification by valence, good or bad, the localization in space, and a directed movement, which of course involves a great variety and a great number of different muscles. And that movement can be toward the prey or away from the predator. Notably, the behavior does not require a veridical representation of the visual environment. It does not require a coherent image or a higher order object recognition. The bird does not have to define the species of fish. The bird does not have to distinguish individual fish from each other. The fish is not interested in identifying the species of bird. The fish simply has to escape a looming shadow. So I'm emphasizing this because now I'm asking, how does this work, and how is kind of that textbook version of how the visual system might do it, and why I think our textbook version is wrong. So a cartoonish view of the visual system that you find in textbooks, in reviews, in systems neuroscience papers is the following. There's assumed to be serial processing of visual information from usually people cite the retina, projections to the thalamus, so the LGN nucleus, and from the LGN to V1. And 20 years ago, people felt the LGN was simply a relay station and all the interesting stuff was happening in visual cortex, V1. That has changed a bit, but still this view of a serial processor still permeates the scientific literature. There's also the idea of hierarchical processing. So the retina is often treated, maybe not by this community that is listening currently, but in many, especially the primate literature, as kind of a camera that does a pixel by pixel representation of the visual image. And then it sends this relatively crude pixelated information to the LGN and then to the visual cortex, where it's then pieced together and gives rise to the representation of features, such as orientations, direction of motion, size, color, etc. There's also the idea that you find quite often is the idea of parallel distributed processing. So it is assumed that the cortex encodes information by means of distributed population vectors. So combinations of neuronal ensembles encode the information about the visual scene. And this is in a kind of a weird twist of science history, I think was taken from computer science, from the idea of deep neural nets that was first developed in the mid-1980s by McLennan and Rumelhart. And it has kind of found its way back to the visual system literature. And there's also a very vocal camp that claim that the visual system is concerned with the coding of image information in an efficient way. And of course, that started in the 1950s, very much influenced by information theory. And Atnev is kind of the very first one who applied this idea of efficient coding to the visual system. And then Horace Barlow proposed 3 principles, one of which was the efficient coding. The first two are, I think, undoubtedly true and uncontroversial. The efficient coding, I think, is still up for debate. And then finally, there's also kind of the view of complex circuitry being in the brain, being shaped by sensory information, by during development, at least by activity or patterns of activity, and by experience. And this is subsumed under the term of self-organization. So I will provoke you all today, and I'll tell you that I think all of these ideas are probably wrong, or at least misguided. Or maybe one could more kind of leniently or graciously say they apply to certain specialized visual systems, but they're not generally true. So on the right, I'm showing here sort of this cartoon version of the rodent visual system, starting in the retina, transferring or conveying information to the LGN, and from there to the visual cortex. And this, of course, on the right is then the picture that has been advertised in the primate visual system. And so if we think about the primate visual system, which has, of course, influences our thinking and is very important to us primates, this is the view that's been proposed by Fellman and Van Essen's important work, really heroic work of tracing the connectivity in this middle part of this schematic, which is all cortical areas that have been implicated in visual processing. If you pay attention, you see here at the bottom, this is the retina with two output channels, the M pathway and the P pathway. And here's the LGN, also an inconspicuous gray box. So the information goes through channels, through LGN, all the way to where the interesting stuff happens. Now let's turn to zebrafish. So I showed you a bird catching a fish, and now the tables are turned. This is a little fish larva, a zebrafish larva, just 0.5 centimeter in length at this age, maybe a week of age. And Duncan Mearns in the lab has recorded with high speed, with high temporal resolution, these prey capture events. So what's indicated here in this red circle is a paramecium. There are other paramecia swimming around. The fish chooses and aims at one of these prey items and catches it. And this is actually the same fish filmed from a different from above, from a different angle with a different camera. So you can see that the fish sneaks up on the prey, likes to come from below, and then opens the mouth and sucks the prey into his mouth. And a hungry little fish larva can empty a dish with about 100 paramecia in an hour. So here's just a still showing the last moment in the life of a paramecium as it disappears in the gaping mouth of its predator. So Julie Samohag, when she was in the lab, pioneered the imaging of brain circuitry during behavior. She developed this paradigm where she mounted zebrafish in agarose. So here's a little fish larva that's embedded with its head in a slab of agarose. You can see actually the border of the agarose gel here. The tail is still free to move and tells us about the movement, direction, and also the intention of the fish. In a variation of this assay, we can also free the eyes and observe eye movements and jaw movements in this kind of preparation. So Julie did some psychophysics by showing different types of stimuli to the fish to determine which ones would evoke prey capture. Here, she was just having fun moving with the mouse, the little white dot around, and could evoke these prey capture behaviors in this head-fixed preparation. You can even see the so-called J-turns, which are orienting movements of the tail, which would, if the fish were stuck in agarose, lead the fish larva towards this assumed prey. And she did psychophysics, systematic psychophysics, this, and found that in this preparation, a fast-moving 5-degree spanning white dot on a dark background was the most efficient stimulus to elicit hunting behavior. Now, when Julie used a different stimulus, such as a black disk on a white surface that, I'm going to play this again, that was expanding in size and displayed on the side, she could elicit an entirely different behavior, which was a very vigorous escape maneuver, which would, if the fish weren't stuck in agarose, would have propelled the fish away from this approaching object on a collision course. So with this kind of paradigm, we can do imaging, because now we can place the behavior on the stage of a 2-photon microscope. And with GCamp, transgenic fish, we can observe the activity in the brain. And we'll show you several examples of this in my talk. We can also do optogenetics by injecting light via a laser into the brain and activate certain populations of neurons. So let's just take a very short detour and tell you and confirm with you that the retina of the zebrafish looks like a regular vertebrate retina. There's a photoreceptor layer that passes on via synaptic connections the visual information to a layer of interneurons here. We have horizontal cells, bipolar cells, amacrine cells, and then, of course, a row here in the ganglion cell layer of cells that receive synaptic inputs from these interneurons and then send long projections across the midline of the brain into visual centers. So we were interested for a few years now in how the central visual brain areas are organized. And Estuardo Robles in the lab carried out a heroic series of experiments where he labeled hundreds of individual retinal ganglion cells, determined their patterns of dendrites in the retina, and for this very same neuron, also traced the connections into the brain. In this case, the retinal ganglion cells is a bit of a diffuse arbor. We don't see the axon because it's deep in the brain, crosses the midline at the optic chiasm, and then dives up to project here to the optic tectum in the zebrafish midbrain. And here it arborizes in one of the tectal layers. And Estuardo collected close to 500 of these images. We now have close to 1,000 retinal ganglion cell morphologies. And he then classified the shapes of these retinal ganglion cells by their dendritic morphologies. And he found sort of the typical assortment of retinal ganglion cell shapes that you can also see in chicken or in mammalian retinas. So we have monostratified types that arborize their dendrites in different layers of the inner plexiform layer in the retina. We also have bistratified and diffuse types, and also funky types like this one here, which reminded us a little bit of a girl in a dress upside down with a very long foot. So this is actually a kind of a very funky zebrafish specific retinal ganglion cell type. And then also this one here, which is interesting because it actually reaches out to the outer plexiform layer and receives direct input potentially from the photoreceptors. So this is where the photoreceptors form synapses on horizontal cells. So here is a schematic depiction of all the shapes that Estuardo could see in the retina. This is probably a very conservative list, schematic. So conservatively, we find about 14 dendrite stratification patterns, which is in the same range as shown in other vertebrate retinas. And just to remind you, the idea behind these different shapes of retinal ganglion cells is that each of these represents a type, and each type serves as a feature detector. So each type responds to one aspect of the visual scene, such as off or on information, edges, chromatic information, motion, direction of motion, etc. And all of this is then combined and sent through the optic nerve into the brain, where it is then used to analyze the visual scene. So this is, of course, not our work, but work from many labs, including the Euler, Baden, Meister, Sainz, Maslin, Westland, Roska lab. I'm probably forgetting some important contributors here. So, but I told you that we can also, for the very same neurons for which we have their dendrite shapes, we can also determine where in the brain they are projecting. And here, Estrado could identify 20 axon projection patterns. So he recorded them individually and found some simple ones. So for example, in this retinal ganglion cell just arborizes in one layer of the tectum. This one is a bit more complicated because it makes a collateral here in an arborization field in the pretectum, so-called AF7. And then we have all kinds of shapes, as I said, 20 different combinations, all the way to very complicated ones. This is the record holder with collateral in the hypothalamus, in the thalamus, in the pretectum, so in one nucleus of the pretectum, and here in the deepest layer of the tectum. Now, when Estrado counted up all the combinations of dendrite and axon shapes, he came up with 75 what we call morphotypes. So these are specific shapes of or types of retinal ganglion cells as defined by both their dendrite and their axonal patterns. So and the notation here is such that here's the cell body, and then these are the layers of the inner plexiform layer to which dendrites are then projected. And these indicates the different pretectal and thalamic areas, and these are the tectal layers. And you can see all of these different combinations are encountered in the zebrafish brain. However, counting more of them and registering more of these ganglion cell shapes doesn't give us more shapes, so we believe we have saturated the system, and there's not going to be more than 75 or so of these types. So the conclusion is, so unlike and sort of deviating from the textbook view, there's in fact very many more than 2 channels that originate in the retina. We find 20 parallel processing streams that originate in the retina of the zebrafish, and these different types of retinal ganglion cells are indicated here in different colors. They project to 9 different targets here in addition to the optic tectum, which receives the majority of the input. And these pie charts are meant to indicate that different dendrite shapes or different dendrite types are combining their projections in each of these arborization fields in the brain. So I don't have time to show you all the experiments. Suffice it to say that for a handful of cases, we've also shown defined function for particular processing streams. For instance, we identified populations of retinal ganglion cells that project into AF7 here. And these retinal ganglion cells respond to small motile objects. They represent the frontal visual field. The AF7 is organized retinotopically. That is, there is a miniature map of visual space in this area. Of course, there's a big and prominent retinotopic map in the tectum, but there's also kind of a little outstation here of the map here in AF7. And this area, as we've shown with laser ablation, is necessary for prey detection. Isaac Bianco's lab a few years ago showed that optogenetic activation of neurons that receive input from AF7 could actually elicit parts of the hunting behavior. So we believe that this is a processing channel into AF7 that is important for prey detection and for the initiation of hunting. And these are the two types of retinal ganglion cells that project to AF7. There's just two of them, a bistratified and a diffuse type. And these are these guys here. And we don't exactly know what their individual function is, but one cool idea that we are currently investigating is whether maybe one of them may encode speed and the other the size of the object. And together, these two features may confer prey identity to a visual object. But we are still working on this. So this is currently a working hypothesis. So do the morphotypes also correspond to molecularly defined cell types? And for this, a few years ago, Yvonne Kölsch in the lab, a very talented graduate student, teamed up with the lab of Josh Sains and his postdoc Karthik Shakar, who now runs his own lab at University of California, Berkeley. And Yvonne took advantage of single-cell RNA sequencing approaches and sequenced the transcriptomes of tens of thousands of retinal ganglion cells isolated by fax sorting. And when she did that, she could, using dimensionality reduction, she could identify 32 different types of retinal ganglion cells indicated by these clusters. So each of these dots represent one cell, the transcriptome of one cell. This is plotted here in 2 dimensions to allow visualization of these clusters. And many of these clusters are very small. And then there's also one cluster that encompasses about 30% of the retinal ganglion cells. And so we now have a pretty good estimate of the number of molecularly defined cell types, which is less than the morphotypes. I should mention that when you look closer and recluster just individual of these clusters, then we find further subdivisions. And when we do so, we end up with about the same number of molecular types as with morphological types. A nice kind of side effect or a nice product of these kinds of sequencing approaches is that we can identify molecular markers for each of these clusters. And many of these markers are specific to particular types, and those can be then the starting point for making a transgenic line that gives us genetic access to individual types. So 4 of these lines are shown here that Yvonne generated using CRISPR-Cas9 genome engineering. And it is very hard to see the retinal ganglion cells. Here you can see a few. This, of course, is the tectum to which these ganglion cells project. But as is common for for pretty much all of these transcription factor markers, they also are expressed in other subpopulations of neurons outside of the visual system. So Yvonne needed to resort to an intersectional genetic trick to label just the retinal ganglion cells in these populations. And she took advantage of a combination of the Q system, which for those of you who are not familiar with this system is similar to the GAL4 system, GAL4 UAS system, and the Cree's recombination system. So when she used a pan-retinal ganglion cell Cree line and crossed this into a type-specific QF2 line, then the Cree would cut out this cassette in our QUAS cassette, would cut out anything that's between the LOXP sites, which is the GFP, and the resulting cells, which are the retinal ganglion cells, would then be labeled red, while everyone else who's not a retinal ganglion cell would remain green. So here we find the muscles are still green, the cells in the hindbrain are still green, but the retinal ganglion cells here in the retina are now colored red. So we can do this now on a decent scale and investigate individual populations of retinal ganglion cells using this approach. And when Yvonne did this here for 5 different lines, she found that each population of retinal ganglion cells, for instance, here is the MAFAA type, project into subsets of the retinal targets. This one labels AF7 and the superficial layers is the tectum. Here's one that just labels one SFGS, so middle layer of the tectum. Here is a type that labels actually a mix, probably several types that project into these hypothalamic and thalamic nuclei in addition to the deepest layer of the tectum, etc., etc. So I think we have now genetic access to each of the subpopulations of the retinal ganglion cells, and a lot of work still awaits us, and everyone is welcome to check out the catalogs of lists that we have published. So here's an example of one line that Yvonne created, which labels this the EOMS-A subpopulation of retinal ganglion cells. These are enriched here in the ventral retina. And when Yvonne ablates these cells, the fish is unable to show phototaxis. So they no longer prefer the light side of the dish. And we also, of course, know the projection patterns which go into these pretectal areas, AF4 and the thalamic area AF4, which by other researchers before have been implicated in phototaxis. So these are candidate prey detector cells that express the transcription factor MAFAA. This transcription factor is expressed in the 2 subpopulations that I've indicated before. Unfortunately, MAFAA is not terribly clean. It's also expressed in other subsets of retinal ganglion cells. And we are currently trying to use genetic tricks to isolate this population of cells to do the experiment similar to the phototaxis experiment that Yvonne did by ablating these cells or manipulating them to show that these cells are in fact involved in hunting. We were more lucky with this one here, which is where we have one transcription factor that labels just one type of retinal ganglion cell. So the picture that has emerged is that of a combinatorial processing of feature-selective ganglion cell inputs. So we find 32 different ganglion cell types that project into these different areas of the brain, including the layers of the tectum here shown on the right, and form 20 different channels into the visual brain. And each of these feature detectors, or several feature detectors, are often combined in individual areas. So one of these, what I dare to call a labeled line, is responsible for prey detection and for initiation of hunting. These retinal ganglion cells come in 2 flavors, and they project into AF7 and into this most superficial layer of the tectum. Another line is important for detection of looming. There is a complicated system that detects off or dimming of the entire visual scene. There's a phototaxis system here in the pretectum and thalamus. There's also a direction of motion dedicated line that forms a collateral in AF5 in the accessory optic system, and then continues to arborize here in this particular layer of the tectum, and then and etc. and so forth. So ongoing work is going to, or future work is going to probably map out the entire visual system and assign functions to each of these channels. Okay, so there are parallel channels for what you may call visual reflexes. Okay, but what about real vision? So what I mean by real vision is things that maybe machine vision people might be interested in. How can we detect and categorize objects in space? So the area in the brain that is probably, that's the best candidate for doing these kinds of things in the larval zebrafish visual system is the tectum. The tectum is the largest area. It receives input from more than 95% of the retinal ganglion cells, often only by collateral, but okay. It has a complicated architecture with different layers that represent different features of the visual scene. It has a beautiful retinotopic map, so a complete representation of the visual environment. So it is in a position to now do interesting things by distributed coding, by population activity, and might encode in complicated distributed ways visual objects. So we did an experiment to test this hypothesis, to give distributed coding some opportunity to show itself. So for this experiment, Dominic Förster in the lab created a triple transgenic line where he expressed cytoplasmic GCAMP in the axons of retinal ganglion cells. So this is here the neuropil of the optic tectum, and these bright structures are the axons of the retinal ganglion cells that are very densely innervating the neuropil of the optic tectum. And in the same fish, he also uses a nuclear localized GCAMP that is now restricted to the cell bodies of the tectal neurons. Okay, so and now by simply by recording in a fish that has both of these transgenes, he could, in the same fish, determine input and output relationships of tactile processing. Okay, and this is of course now done on the stage of a 2-photon microscope, and via a projector we can project different stimuli to the zebrafish. The battery of visual stimuli that we've used was quite diverse. All the things that we could think about. A dark ramp, meaning a slow change in ambient illumination, dark and bright ramping, also sudden flashes of darkness or of brightness, prey-like stimuli, larger moving stimuli, as well as grating motion, looming, etc. So when Dominic hierarchically clustered the responses of the tactile neurons to these diverse stimuli, which are plotted here on the left, he found that there was a group of cells that was very specific and had a constant response to small, either stationary or moving stimuli. So they have the strongest responses to these kinds of stimuli that are shown here. 5-degree dots moving in 2 directions. And were silent when the whole visual field was changed in luminance, and responded only weakly, and only some subclasses responded to some other stimuli. So they were quite specific to small moving objects. Another group was quite specifically responses to looming and dimming stimuli. And they were really not in this clustering, not related to the ones that responded to small moving objects. And then in the middle, there was some so a large group, maybe half of the tectum, that responds to some intermediate sizes and might respond to close objects or approaching objects. Okay, so here is just a pie chart that shows different classes. So this is a simplified schematic of what I just showed you. So the most frequent classes of retinal ganglion cells. These are the ganglion cell responses, and then these are the tactile neurons. And Dominic, with the help of Thomas Helmbrecht in the lab and Joe Donovan, investigated how you can transform the retinal ganglion cell population responses to the tactile population responses. And the answer was actually quite shocking, in that most of the tactile responses could be explained by a linear combination of the retinal ganglion cell inputs. There's a little bit that the tectum contributes. It introduces more directions and direction selectivity to some of the channels. But actually, the contribution of the tectum for the sensory encoding was quite limited. So Dominic was interested in whether the tectum was organized in such a way to represent a prey object in retinotopic coordinates. And he was starting with sort of a simple assumption about the hunting episodes. What happens to the visual image as the fish is initiating a hunt? Now, in the typical case, the fish will first detect an object here in its peripheral visual field at a distance. So it will appear small and will be represented in the nasal, by the nasal retinal ganglion cells and in the posterior part of the tectum. Then the fish will detect this and categorize it as prey and turn towards it to bring the prey here into this binocular field of view right in front of it. There it will, of course, be much larger because the fish is now closer, and it will be represented in the anterior part of the tectum, and actually in the anterior half of both tecta in the binocular zone. So during a hunting episode, there will initially be a small object here in the posterior tectum that then moves across the tectal surface and will be bigger and be represented in the anterior tectum. So the cells that represent these kinds of objects distributed in this way as predicted by the sensory ecology? And the answer is resounding yes. The back of the tectum is enriched with cells that respond to a small moving object that is actually moving. Then there is a sensitivity to small objects all over the tectum, but then in the anterior part of the tectum, the responses to the larger objects are enriched. This is also shown here in the population distribution. This is the axis of the tectum, and you can see this very nice peak of cells where cells that are responding to small moving objects are enriched. So this, of course, now recapitulates the sequence of hunting, as I've indicated before, and very nicely matches how the image is developing during prey capture. And I'd like to give a shout out here to work done in Tom Baden's lab on asymmetries in the zebrafish retina that has also discovered asymmetries in the arrangement of both photoreceptors, but also retinal ganglion cells in the zebrafish retina, which also presage or beautifully match the expected sensory ecology. And it will be very cool in the future to link these different patterns in the retina to those in the tectum. Okay, so this is kind of the fate of even of the most beautiful works of PhD students that 5 years of work are summarized in just 1 or 2 slides when their PI gives a talk. This is a summary of what Thomas Helmbrecht, a super talented student, did in the lab. Thomas was interested in moving, in understanding the sensory motor transformation that was going on in the tectum and identifying the output channels of the tectum now. The tectum is connected to areas in the hindbrain and in the midbrain tegmentum, and of course steers the movements of the fish. So he was interested in whether he could identify particularly dedicated channels that were specific to the behavior of the animal. And the answer is yes. So here's the summary of his thesis. He identified 2 bundles of axons that originated in the tectum. One was dedicated to escape, avoidance responses that projected here along these medial tract of this tectobulbar fascicle. When he activated these cells, the fish would make a very vigorous tail response away from the eye that was connected to this tectum. So this was done using optogenetics. And he could also show with calcium imaging that this tract of axons responded to a looming stimulus. And another group of axons here in this lateral region of the tract was activated when the fish was seeing a prey-like stimulus. And in fact, there was a topography among these axons in that the further lateral you came, the more, the greater the amplitude of the tail bend was that he could elicit with using optogenetics. So what we conclude from this is that the connections from the tectum to the hindbrain are organized according to a space code, importantly not a rate code, as many people would have predicted. Distinct labeled motor lines exist for approach and escape. Okay, so I think I've shown you, I hope I've convinced you that the zebrafish visual system is organized in a way that contradicts or at least challenges the textbook view of how the visual system of a vertebrate might be organized. Now let's kind of challenge another dogma of the field, namely that of the self-organization of neural circuits. So many years ago, Linda Nevin, when she was in the lab, performed the following experiment. She exposed the fish to different light regimes. So this would be the normal light-dark cycle. She kept the fish in the dark. She changed the balance of on and off by blocking the on channel, or she actually inhibited and blocked any synaptic transition using botulinum toxin. And she could not, in these experiments, find any difference in the layering of retinal ganglion cell projections into the tectum, which led us to conclude that, at least for layer-specific targeting, experience or even retinal activity, synaptic activity, was not necessary. But we wanted to revisit this issue now with better methods, molecular methods. So Shahar Sharman in the lab, a talented graduate student, decided to use single-cell RNA sequencing to develop to survey the cell types in the visual forebrain and also the tectum of fish that have never seen a ray of light through the retina. So this is just this is the single cell RNA sequencing data from 100,000 cells from this is done from a transgenic line that labels pretectum and thalamus and hypothalamus. And he finds in this collection GABAergic neurons, glutamatergic neurons, about 40 or so cell types each. And he even finds progenitor cells, precursor neurons, etc. And then also the habenula is part of that pattern also. And here's just the GABAergic and the glutamatergic population. And beautifully, he can also follow the differentiation path from the progenitors. So these are still dividing. And then there is a neurogenin 1 path that leads both to the habenula and to the glutamatergic neurons. And then also a developmental trajectory to the GABAergic neurons, which use the transcription factor ACL1B. And the habenula then has its own kind of private path that uses both neurogenin 1 and the chemokine receptor CXCR4B. Now, when you look in Lakritz mutants, which are devoid of all retinal ganglion cells, so these are fish that look normal. They are dark because they're unable to do visual background adaptation. They lack all retinal ganglion cells because they have a mutation in a transcription factor that is important for the cell fate determination of the retinal ganglion cell lineage. So, and then do the same kind of single cell RNA sequencing in the same line. Shahar found no major difference between the wild type and the Lacritz mutants. And I don't have time to show you all the statistics that he did in order to detect any differences. He basically could not detect any cell type that was missing or not induced in the Lacritz mutant in the forebrain. And he did find individual genes that were up or down regulated, but the cell fates themselves were unchanged. And even the proportion of the cell types was unaltered in the Lacritz mutant. So this was very provocative for us because, of course, there's a huge literature on how visual experience and activity shapes the neural circuits that are downstream of the retina. So we asked, well, can we maybe find the deficit in the behavior itself? Now, of course, these Lacritz mutants lack all retinal ganglion cells, so they don't respond to any visual stimuli. They are completely blind. So we resorted to optogenetics to elicit an optokinetic response. This is based on work that Fumi Kubo did when she was in the lab, which was recently picked up by Yunmin Wu, a graduate student in our lab. So what you do here is you put the fish in an arena, and a wild-type fish, when exposed to a moving grating that moves around the fish, will respond with optokinetic eye movements, which are probably familiar to all of you. And Fumi and Yunmin now managed, by placing an optic fiber near the accessory optic system and using a channelrhodopsin transgenic fish, to elicit optokinetic eye movements. You can see the two eyes moving here in this animal. So this is notably without any visual stimulation. So we can evoke optokinetic responses in this animal without any visual stimulation. Now, the question was, can we do the same in Lacritz fish? And the answer is yes. So here is the OKR index plotted for a wild type and Lacritz genotype. When there's no stimulation, there's no optokinetic response. When there's a visual stimulation, only the wild type response here with a strong optokinetic response. And when we do an optogenetic stimulation, then both Lacritz and wild type show these typical eye movements. So this suggests that the circuits that evoke these eye movements are assembled properly in a fish that has is not innervated by retinal ganglion cells. Okay, so let me summarize. Oh, I need to wrap up. In what way is the view of vision changing? So the visual system of larval zebrafish employs matched filters at each stage of processing. There's no such thing as a pixel-wise representation of the visual system, visual seen by the retina. Retinal ganglion cells are single feature detectors. The downstream neurons then receive inputs from a combination of retinal ganglion cells. And in the pretectum and tectum, we observe combined feature detectors. The 20 or so visual pathways resemble labeled lines. They are often dedicated to particular behavior outcomes. Attractive and aversive stimuli are processed in parallel segregated circuits, so not parallel distributed circuits, but parallel segregated circuits. I did not have time to show you that there are circuits that ensure that motor commands are not in conflict with each other. A fish cannot approach and avoid at the same time. So this is done by interneuron populations that inhibit in a reciprocal fashion different lines. Elementary forms of spatial attention, also I didn't have time to show you that, are implemented by circuits that are dedicated and that suppress responses while enhancing responses to the most salient stimuli. Then finally, I showed you that cell types, layers, connectivity in visual brain areas are assembled by activity-independent genetic programs. And as an aside, maybe for those of you who don't care about zebrafish, so it's just something to think about. So why is biological intelligence still vastly superior to artificial intelligence? So the problem is that even the most sophisticated deep neural networks fail at simple tasks. They need very large training sets that need to be labeled to train the net. And they're also very inefficient in terms of their energy consumption. The possible reasons, so those are maybe more points for discussion, are that machine learning ignores the source of innate knowledge that our brains have. Machine vision is trained to classify images, to name objects, and to recognize faces. Advanced systems may estimate agents' behavior. These are all very human-centric tasks. This is not what most visual systems care about. Brains have been selected to survive by responding properly to external cues. And for this, they don't need a veridical representation of the external world. This by itself does not confer any advantage to the animal. Brains are so efficient because they use the energy spent by development and learning over the lifetime of the organisms and indirectly over evolutionary timescales. And then something that I think the AI community also begins to realize is that the brain assigns meanings to stimuli. So, and we don't know how those are encoded, but clearly brains have found clever ways to develop some semantics. So the common claim that deep neural nets are biologically inspired can only be justified if you believe that common misconceptions of the visual system, I mentioned serial, layered, hierarchical, parallel distributed, self-organizing, are accepted. Okay, with that, I'd like to just show you a few more slides to advertise our atlas of the zebrafish brain. I showed you a lot about the anatomy of the brain. What I did not mention or did not emphasize is the small size. So this is the larval zebrafish brain in the bottom left compared to that of the adult mouse. So any connectomic or functional neuroanatomy project is going to be much more daunting in the mouse. So we've, over the past couple of years, developed atlases of the zebrafish brain. We've mapped lots of individual cell morphologies into a standard reference brain. These are the tactile interneurons. These are the tactile projection neurons mirrored to the other side of the brain. And we have, over the years, collected many more morphologies of neurons and are now filling the entire central nervous system of the zebrafish with really thousands of morphologies. This is a kind of a pipeline in the lab that we are using. And check out the atlas at the website that is given, and you can find the shapes of neurons in your favorite area. And you can actually also determine where these areas are receiving input from and what its outputs are. In the same space, we've also mapped now over 450 markers, most of them transgenic lines that you can also check out and pick and choose and go shopping for. And we are now adding, this is in preparation and not published yet, also gene expression pattern using HCR in situ hybridizations, which are super sensitive and can be multiplexed. And we are adding this information about gene expression also as we speak. Okay, so with that, I'd like to finish up and I'm happy to take questions. Well, most important slide is this one here. This is, oops, oh, I should. This is the group of superheroes that have done all the work. This is in the happier pre-corona times at one of our lab parties. And I mentioned the individual people as I showed the data that they've contributed. With that, I'd like to thank them and thank you for your attention.
Thank you very, very much. It was a really great talk. I really enjoyed it. And I can see here that you haven't lost a single viewer during the whole hour. So, I mean, there was so much in there. It's incredible. I see there's some questions. One question that I had, I think you mentioned that in the summary slide, but maybe you could say one or two sentences more to it. The real environment of the larva zebrafish, I guess, is very confusing because there are all of these stimuli at the same time, like different paramecia swimming around. Maybe the light becomes darker, some spot comes closer. So do you think that this might be the really important role of the detectome, that it kind of decides which stimulus is actually the most important at that very moment?
So that is certainly, I wouldn't call it a function of the detectome, but I think it's a necessary, task, or it's a necessary feature of processing. So the function of the detectome in my view, which is actually conforms to the classical view of the detectome, is that it transforms a sensory map, a visual map, into a motor map. So this is what all these complicated, this neuropil connectivity is for. And we don't understand how this transformation works, right? We know inputs and we know the outputs, thanks to Thomas Helmbrecht's work, but we don't know how these two maps are converted into each other. This is a fantastic problem. But of course, in order to choose one visual target and initiate the appropriate behavior, you need to ignore lots of other stimuli.
Yeah.
So I want to refer you to one paper that we've published recently that showed that for stimuli that are presented to the same eye, it is in fact already probably in the retina that there is a lateral suppression. So an enhancement of a cluster of retinal ganglion cells at the position of the most, that represent the most salient object, and a suppression of other responses in the neighboring regions. And we claim that because we see in the retinotectal system, we see this kind of prioritization in the activity of the axons. And it is not dependent on the detectome. So we ablated the detectome, and we just had a detectome that had the retinal axons as an intact sheet, and they as a population still could choose the most salient stimulus. So if you present now the stimuli to the two different eyes, then there's an additional nucleus involved, the nucleus isthmi. And we think, I wouldn't say the case is not closed yet, but all the evidence, the correlative evidence, points to a role in the nucleus isthmi in determining which detectome is going to be favored, which detectome is going to activate the behavioral program, and which one is going to be suppressed.
Okay.
Check out the paper. It came out in Neuro a few months ago.
Yeah, thank you. So I'm just going through the questions that are being written in the chat. So one question is, larvae are quite reflexive in their visual ways, but this becomes less obvious as fish mature. Perhaps what one might call traditional image-forming vision becomes the luxury of the bigger brain.
Yes, I mean, that's a possibility, certainly. We start with the larva because it's small and can be imaged, etc. But of course, the fish grows substantially, and the detectome grows with the growth of the rest of the organism. So it is, I mean, I don't know in volume, but probably 1 million times larger. Maybe that's an exaggeration, but it's much larger. There is room for population coding, distributed coding in the adult detectome. Yeah, I grant that. But at least it shows this visual system at this stage doesn't need that, or doesn't employ that kind of coding.
Another question is, so Emery says, impressive talk. Can you please tell more on ganglion cell innervating zebrafish equivalent of mammalian LGN? Do LGN innervate ganglion cells have features different than others, or rather they have mixed features?
Okay. So LGN, that's a big debate in the teleost field. So this branch of vertebrates, fish, some reptiles, birds, have used as a major way of information transfer from the retina to the telencephalon. They use the tectum. So the projections, so there's a tectothalamic projection. So a visual LGN probably does not exist in the way it does in mammals. Okay, there are 3 retinorecipient thalamic nuclei in zebrafish, but none of them is really projected into the telencephalon. So none of them is a good candidate for the LGN. I was very much in doubt of this, but I've become persuaded by the teleost neuroanatomists who tell me that. There's a nice review article by Thomas Müller about the zebrafish, about zebrafish thalamus, visual thalamus, that makes the point very clearly, I think. So, yeah.
All right. Philip Bartle asks, what do these data mean for perceptual spaces in zebrafish?
Perceptual spaces?
Yeah.
Maybe Philip can.
Yeah, I'm also not 100% sure what
perceptual spaces. Yeah, I mean, I don't know more than what I showed you. We published in the first 10 years of my life as an independent faculty, we published paper with perception and claiming perception in zebrafish, and we got flack from the reviewers that fish don't perceive anything, and we defended that. And now I've come full circle and think maybe there's no such thing as perception in larval fish. So I'm becoming very skeptical of the papers that do whole brain imaging and say, I don't know, chromatic processing in distributed networks in the fish. And I think first you have to show that there's color vision in fish. I mean, there's certainly selective cone usage. So you from the Baden lab know that, right? So certain behaviors use particular color cone channels. But is there really something like color vision like we have? I'm not sure. That's up for an interesting discussion. But yeah, perception is a difficult concept.
Maybe Philip will join the talk.
It's like cognition. Cognition is problematic as a concept.
Yeah, maybe Philip join the discussion afterwards.
I'm looking forward to talking with Philip.
So Leon Lagnado asked to the topic, what happens in optic tectum? You said that you think that responses of neurons in the optic tectum are linear combinations of inputs from the ganglion cells. What do local interneurons do? And then he extended the question a bit, adjust weights, perhaps depend on signals from other senses or changes in internal state?
All of the above. So first of all, I think the tectum, as I said before in response to your question, the tectum has a sensory function, but also a motor function. So that transformation, I think that's what these interneurons do. And the tectum also is important for the saliency encoding that I think also you asked about. The tectum is also important for, mediating the state, right? So we've shown a few years ago that a hungry fish processes prey differently than a satiated fish.
Yeah.
And so hunger recruits more neurons that respond to prey. And interestingly, it does not switch the feature selectivity of the tectum neurons. So, but it wakes up, it wakes up from a quiescent state neurons that would, in a satiated fish, not respond to prey. So the detectome has lots of functions. It does the linear combination is sort of the general rule. There are additional things that, so for instance, Martin Meyer showed very nicely that the detectome has additional direction selectivities. Tectal neurons have additional direction selectivities that are not impinged on or not imposed on them by retinal ganglion cells. So.
Right, I think that's all the questions for now. I will now post the link so you can join the Zoom room. And so I will let this live stream run for a few minutes longer, but then afterwards I will shut that down, and then unfortunately also the chat will disappear. So if you want to join the Zoom discussion, you kind of have to decide now. I think sure. Thomas
so
already joined.
The Zoom link was also in the email that you sent me, right? It's a different Zoom link.
No, it's, no. You just stay where you are. It's just for our viewers. So, for example, Thomas has already joined, and I think some more people will join.
All right. We're still live, are we?
We're still live.
No secrets,
No, not yet.
That was great. Yes. I was particularly struck by one of the very last slides that you had, which showed the in situs. That looked really lovely. Yeah. And then you sort of moved away, so I couldn't really see it properly, but it looked really good.
Yeah, this is with the method that we shared with you, right? With the HCR in situs. Yeah. No, this is very nice. And it took us a while to register these in situ stained whole mounts to our standard reference brain, right? Because you have to do all the this warping. But this works now very nicely. And it's going to be something like 150 or so genes really interesting genes that come out of the SCRNA-seq data that we've mapped to the brain. So there will be a lot of information.
Yeah. No, I mean, I like the idea that these little fish use labeled lines for a lot of their tricks. This is actually something that's starting to be more and more obvious in mice as well. I don't know if you saw the recent paper about Daniel Kirschensteiner, the one where they killed some alpha cells and then they don't hunt so well.
Yeah.
Yeah.
No, I think it is. I think the idea is there. And I'm kind of maybe put a nail in the coffin of the parallel distributed processing idea. I think it's kind of it lingers below the surface of many of these studies. And I think initially the mouse field tried to make the mouse a little monkey, and now it turns out no, the mouse visual system is not really interested in faces, right? But it's interested in escaping or in finding crickets to eat. And
it certainly is. I'm just wondering to what extent it's mutually exclusive. The mouse could still be interested in faces as well as escaping.
Yeah. Well, yeah, maybe that was Philip's question also. You know, where is the perceptual space? Yeah. No, it's true. Yeah, maybe I was. I mean, I was trying to be provocative to make.
No, I mean, I think for the larva,
I believe in it, but you can still be provocative with what you believe.
And, you know, I think for the larva, I think this is probably the right direction. I wouldn't go as quite as extreme as saying there's no general processing. I mean, what you're basically saying is everything specific, right? Cell 1 does this, cell 2 does this, and then once you've done all the cells, then that's all the functions that the fish has.
Okay, you are more lenient, more gracious. I think putting out the strong hypothesis has the advantage that you can refute it, right? With one counterexample. So I'm waiting for that.
Except that general coding is really hard to prove, where specific yeah, it's easy in the sense that you kill the cell, it's gone. What else is it going to be, right? I mean,
Yeah.
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 I guess.
Yeah.
What would the proof be? What would be the experiment that would show that there's what you might, well, I don't know, the term visual processing centrally, interactions between these channels, I guess. Is that what we would need to demonstrate, that interactions, downstream of the retina between channels? Is that, what's the criteria?
I mean, actually, the interactions happen. I went through this too quickly, probably. You still have mutual inhibition so that you don't have conflicts, right? So
right.
Reciprocal inhibition. I think I would accept an experiment that did the following, that you could activate one population of cells, get one behavior, and activate an overlapping, different but overlapping population and get a different behavior. So this is a relatively simple experiment. So that would show to me that, yes, it's a population